Hello, my name is Konstantina Falida, and I'm working at the editorial office of the International Journal of Cancer. It's the first time that I'm attending an EASY conference, and I'm really excited to share with you our study on the importance of cell line authentication in biomedical research. A misidentified cell line occurs through cross-contamination, mislabeling, or other laboratory errors, meaning that it no longer corresponds to the original donor, and it may in fact come from an entirely different species, tissue, or disease. The tremendous damage of the use of misidentified cell lines in biomedical research has been shown exemplarily by a study from Korch and Capes Davis. It was estimated that almost billions of research dollars have been spent on studies using two commonly misidentified cell lines, while at the same time, data based on misidentified cell lines could misguide and delay therapy development, resulting in missed opportunities to improve human health. To avoid publishing studies that are based on misidentified cell lines, the International Journal of Cancer has introduced a stringent cell line policy already since 2010. In the editorial office, it's my responsibility to check all cell line-related issues of manuscripts submitted to our journal before the peer review starts. We request cell line authentication reports of all human cell lines used in the study, listing of the official cell line name and its research resource identifier, and a statement confirming the authentication in the materials and methods section. Last year, we published a study providing an overview of issues pertinent to misidentified cell lines and discussing available solutions. More than 5% of the cell lines used in manuscripts submitted to our journal were misidentified, and the most frequent contaminant was the HeLa cell line. We also found that most of the misidentified cell lines were used in studies for gastric and liver cancer, which indicates that the literature on those cancer types probably suffers a lot from misidentified cell line data. When we look at the manuscript level, then we see that based on the available information, the majority of the selected manuscripts have no cell line-related problems. However, in 130 manuscripts, moderate or severe cell line-related problems are observed. Finally, 35 manuscripts, which is almost 5%, were rejected for severe cell line-related problems, which means that at least 2 cell lines were misidentified. In order to explore whether manuscripts rejected at the International Journal of Cancer with severe cell line-related problems are subsequently published elsewhere, we followed up 106 manuscripts that contained a large amount of data based on misidentified cell lines. In March 2023, 78 of these manuscripts were published in other journals, of which 72 manuscripts still included the data of the misidentified cell lines, although the authors were informed about the cell line problems observed in their manuscript in our decision letters. Based on our experiences, we have several recommendations for journal and publishers to avoid the publication of studies including misidentified cell line data. Journals should consider to enforce the use of cell line research resource identifiers, which can be obtained at the RRAD portal or at the Cellosaurus database, where actually a warning is displayed when a cell line is misidentified. Journals should verify all cell lines used in a manuscript in the Cellosaurus database, where many cell lines are recorded. In addition, journals should implement mandatory cell line authentication and occasionally check the documents so that regular authentication of cell lines will become common practice. Last but not least, action should be taken to correct already published manuscripts that contain data on misidentified cell lines. Before closing, I would like to mention that this study was performed by Nicole Souren and was supported by other members of our journal as well as some external ones. For further information, you can check our paper by scanning the QR code at the right bottom of the poster, or you could contact our editorial office or me personally. Thank you.